Good afternoon. We will reconvene now. Good afternoon. We will reconvene at this point. It is my distinct honor to welcome to the stage our host, Dr. Kanturk. He is rector of this great university and a professor of medicine in the Department of General Surgery. His research is focused on interactions of hormone signaling on surgical outcomes with an emphasis on oncology and endocrinology. So welcome, Dr. Kanturk.
Dear Chairman, dear colleagues, good afternoon. This study is an ongoing study. This is the preliminary results of our study. I will give you some details about the microbiota and then our results, gut microbiota and the breast tissue. As you know, breast cancer is most common cancer worldwide, and 1 into 8 to 10 women will develop breast cancer in her lifetime. Incidence of breast cancer is increasing year by year, but development of diagnostic method and the therapeutic method, that rate even increase, but mainly decrease whenever compared in relatively. The most common cancer is breast cancer for ladies in Turkey. Almost 25,000 new cases detected in 2020. Breast cancer death rate very depending of development levels of the countries, including quality of health services, regularity of health screenings, and the time for early diagnosis and treatment opportunities. Breast cancer related deaths are still high, I told you, but we have many options to treat breast cancer cases. If we detect the breast cancer earlier stage, it is easy to treat, having the cure, as you know. Early diagnosis is so very important for prognosis of breast cancer. This morning I explained that the Hippocrates is the son of this land. Before Christ, he told all disease begins in the gut. It is very interesting, almost more than 3,000 years ago. A microbiota is the collection of microorganisms in a specified location or sample, and it inhabits all barrier surfaces of our organism, including respiratory, urogenitals, digestive, or skin. There are many functional issues related to microbiota. Microbiota is accepted as an organ, including these microbiota's functions include some food digestion, dietary calorific bioavailability, detoxification, energy metabolism, nutrient absorption, metabolism of bile and hormones, xenobiotic and drug metabolism, and bioavailability. I told you at the same time microorganisms can be accepted as an organ because it takes too much place in our body, and I told you there are many functions related to microbiota. Gut microbiota are critical for maintaining the host health as they regulate a multitude of systemic functions as well as the human system, and thus confer a protective role against the pathogens and carcinogens. In the figure, red arrows depict the regulatory impact of the different factors on the host microbiota. Green arrows denote the effect of the gut microbiota on various functions within the host that help to maintain health of the subject. Microorganisms that symbiotically live with humans are increasingly recognized as important game players. The largest collection of these microorganisms is found in the gastrointestinal tract. Microbiome is collective genome of microorganisms. A symbiotic relationship between the host and the microbiota is called the normobiosis. The gut microbiota differs due to a variety of intrinsic and extrinsic factors, including age, race, diet habits, something more, as you see in the slide. Any disruption in the normal microbiome content that is presented as dysbiosis can be rescued in ways that are not harmful to the host and promote the different diseases. There are balance in the body. There are many quantitative changes, many qualitative and metagenomic changes related to the microbiota, and it is related with the cancerogenic development. Human breast contains a diverse and unique set of bacterial components that are different from other body sites depending on the age of person, sample location, pregnancy story, race, something like that. And origin of the breast tissue microbiome is abundantly, additionally, formed from the gastrointestinal tract via nipple areolar openings, nipple-mouth junction through breastfeeding, and or sexual contact. Recent studies suggest that microbiota of women with breast cancer is different from the healthy woman, and that cancer may grow with different responses to development of some diseases and treatments. There are suggestions that microbiota of breast cancer patients is different than that of healthy persons, and the cancer developments also show differences. Several bacterial genes which are capable of producing estrogen metabolizing enzymes. Accordingly, the gut microbiota has the ability to modulate estrogen serum levels. The collection of enteric bacterial genes that metabolize estrogen is called the estrobilome. Disturbance in the microbiota to estrobilome ratio may increase the risk of breast cancer. The intestinal microbiota is also affected by development of the adiposity and obesity. As you know, overweight and obese women have more risk of breast cancer than healthy weight women. There are many risk factors for development of breast cancer. As we know, some is controlled by ourselves, some is not uncontrolled. In recent years, studies have showed that the microbiota may be one of these factors. However, the relationship has not been clearly established. The degradation of P53 is one of the important effects. The first pathogenic bacterium involved in the development of cancer is Helicobacter pylori, as you know, which is classified as a class 1 carcinogen by World Health Organization. This bacterium produces the virulence factor, CAGA, which induces the degradation of the P53 suppressor gene and in gastric epithelial cells and promote the increase of gastric cancer. Although DNA damage alone is not sufficient to cause the cancer, the most harmful type of DNA damage caused by the genotoxins, reactive oxygen species, and ionizing radiation are double-strand breaks. Pathogenic bacteria may indirectly promote carcinogenesis through the generation of oxidative stress. Toxin produces some bacteria, including Bacteroides fragilis, Helicobacter pylori, can activate the human sperm oxidase enzyme, which produces the hydrogen peroxide and other reactive oxygen species, then cause the DNA damage. Estrobilome and androbilome relationship between the microbiota and hormone-dependent cancers such as breast and prostate cancer is studying. The estrobilome is collection of microbial genes responsible for the mechanism involved in estrogen metabolism and therefore circulating levels. Disruption in the microbiota to estrogen ratio may lead to elevated levels of circulating estrogen and their metabolites, increasing the risk of breast cancer. Aim of this study to reveal the connection between the breast cancer and microbiota in Turkish breast cancer patient. By revealing the relationship between the existing breast cancer and gut microbiota, examining the effect of this relationship for development of breast cancer and other factors affecting this relationship. As a result of this study, if these relations are clearly proved, there will be new studies to show how to prevent breast cancer or to develop early diagnosis and treatment processes. The study approved by ethical committee of our university planned prospective and randomized study for patients operated in our Department of General Surgery for breast cancer. These are inclusion criteria, the age 18 to 65, newly diagnosed early stage breast cancer, and the patients had not received any antibiotic or systemic steroid treatment for last 3 months, and no other known history of malignancy and pregnancy and breastfeeding history, and a number of the cases decided with G-Power program, and 22 patients were recruited for study. After all patients diagnosed breast cancer, they are hospitalized and operated. They were given detailed information about disease and the study, and informed consent were obtained. Height and weight measurements detected, and the 20 cc venous blood was taken from all patients to study serum markers, separated into serum and plasma and Buffy-code, and processed. Stool samples were collected from all patients who informed how to give the sterile stool sample. The breast tumor tissue were preoperatively separated as pathology specimen for frozen examination, and in a sterile environment, the necessary conditions were taken into consideration, and 1 cubic centimeters breast tumor and normal tissue taken and stored 80 degrees centigrade before analysis. Pure link microbiome DNA purification kit was used. The tumor size detected and tumor characteristic collected. These are demographic data of our cases. There is no statistically significant relationship was detected, and bacterial genus, family, and species were analyzed separately from normal breast tissue and tumor tissue and the stool. 22 patients were coded normal, tumor, and the stool samples, and named NTM. 267 genus, 164 families, and 476 species were isolated from these samples of the patients, and their numbers have been determined. Diversity of families isolated from patients' stools, and genus diversity of the patient's stool also coded. Of these isolated bacteria, especially 22 genus, 22 families, and 26 species were found to be present in statistically significantly different numbers in tumor breast tissue and normal breast tissue. This is the comparison. There is a significant difference when compared the stools, but whenever we compare the normal and tumor tissue, we detected, especially in breast tumor tissue, Ruminococcus, Eubacterium, Actinobacter, Stenorophomonas, and Bacillus genera were found in the significantly increased. In the literature, followings are detected. Addition to these: Lactobacillus, Staphylococcus, Bacteroides, Protobacteria, and Propionibacterium genera. These are, as you see, especially Ruminococcus and Actinobacter, different than normal tissue at the tumor tissue. As you see, genus, the red one is positive bacterium for tumor tissue, and the greens are the normal tissue. On the other hand, whenever we analyze the bacteria families, it was determined that especially Bacteroides massiliensis, Bacteroides uniformis, Staphylococcus hominis, Stenotrophomonas, Rhizophilia, and Staphylococcus varnei families were almost completely absent in normal tissue, but they are abundant in tumoral tissue, but were statistically significant. And other bacterium families were found to be statistically significant, more abundant in normal breast tissue, as you see in these table. Family and family names, red and the green one, yellow ones seems very different. And whenever we are compare the species, there is also, whenever we compare the species in 3 samples, there were statistical significant differences, but most often detected species is Burkholderesia, Clostridium, Bacteroides species. These are statistically significantly increased in tumor breast tissue. In normal breast tissue, you can see the other bacteria species were detected. This is also show which bacterium as a species seems important. Most of them seen much more in normal tissue. This table shows the tumor tissue and normal tissue levels. The 22 samples taken in our study show significant differences in the number of some of the features detected, especially between tumor and normal breast tissue. However, comparison of the stool samples between the healthy patients and control groups could not be made clearly. No significant differences were detected between the patients' demographic data. The relatively limited number of our patients may have been effective in these. This study we conducted to reveal the relationship between the breast cancer and microbiota provides important information in diagnosis, follow-up, treatment, and prognosis of breast cancer. It is clear that better understanding of microbiota will provide us with more important information about what can be done to prevent breast cancer before it occurs. Even though our study was conducted on a limited number of patients, significant differences were detected. This has shown that the similar studies to be conducted in larger patient groups will provide us with much more detailed information on this subject. We will, in the future, the study I told you, ongoing study, in near future, we will report the data with different molecular type breast cancers, microbiota, gut microbiota, and tumoral microbiota differences. Thank you very much for your patience.
Thank you, and congratulations, because to develop such a robust data set in a human condition that's obviously very relevant takes a lot of work, a lot of time and effort, very cutting-edge techniques as well as thoughts. We have time for one question, but I'm going to sneak in one, in that certainly chemotherapeutic agents probably dramatically affect your microbiome. And looking at this going forward, you might have an opportunity to see which chemotherapeutic adjuncts to the surgery, also how they interplay with the microbiome. Any comment?
If you look to literature, you can find this kind of studies that the chemotherapy changed the barrier of gastrointestinal mucosa, you know.
Yeah.
So it causes differences in microbiota, and it also affects the result of the chemotherapy selection. We can do similar studies in the future. Maybe we can collect the samples and we can study it.
Yes, you know, as we learned this morning, that what you eat actually impacts your response to chemo, your microbiome, etc. So, any quick question from the audience? Yes.
Yes, you are oncologist.
Thank you very much for your excellent presentation and for this study. So I was curious to know if there is some evidence that some of the bacteria that you found to be enriched in breast cancer tissues when compared to normal tissues also affect tumor response to treatments, because, for instance, in patients undergoing preoperative chemotherapy or chemoimmunotherapy, there could be the chance to use concomitant antibiotic treatments to eradicate Ruminococcus, Acinetobacter, Xenotrophomonas, and so on. I was curious to know if there is also some therapeutic potential for targeting those bacteria in combination with anti-cancer treatments. Thank you.
I think so. We don't need to use the antibiotics for these microbes, but I believe that I told you there is a relation between the selection of oncologic treatment and the success of oncologic treatment. So we will look forward to show the effects of it in our coming studies.
Thank you.
Thank you.
Thank you very much.
Thank you very much.
Next, I introduce to the podium one of my partners at the Brigham and Women's Hospital in Boston, Dr. Eric Hsu, who's an active clinical surgeon. He's also an educator as the Associate Program Director of our Minimally Invasive GI and Bariatric Surgery Fellowship. Dr. Hsu has been educated completely within the Harvard system and only did a short stint studying immunology over at Oxford University. And he's going to talk to us about diet and microbial interactions in metabolic bariatric surgery.
Thank you so much, Keith, for the nice introduction, and thank you, Professor Santork, for the warm welcome here and the invitation to speak. These are my disclosures, which aren't relevant. This is what I'm going to talk about today. I think bariatric surgery is a little different from the rest of this conference. I'll give a quick overview of that just to set the stage. I'll talk a little bit about the diet and microbiome and how they interplay, particularly with the metabolic effects of surgery, and highlight some of the work my lab has done and some of the ongoing work, if we have time. But I will try to keep on schedule here. So bariatric surgery, these are the two main types. These are all surgeries that either rearrange the intestine in the stomach with the gastric bypass here on the left, or with the sleeve gastrectomy, which is the most common surgery done now internationally. You simply remove this portion of the stomach. So very simple to conceive operations, really designed by surgeons originally thinking, we will make the stomach smaller, or we will divert food so it skips part of the stomach, and therefore you will lose weight because you eat less and absorb less nutrients perhaps. These work very well. This is from a randomized control study in Scandinavia of sleeve versus gastric bypass, looking at change of body mass index over the course of 5 years. And on average, after a sleeve gastrectomy, you lose about 25% of your starting weight, or 10 to 12 BMI points. Bypass, you lose a bit more. And over the long term, this is maintained in the average patient, though you can see there's a lot of variability over time. Really importantly, this is not only a good treatment for obesity and for weight loss, but this is the best treatment there is for type 2 diabetes. And this was shown most well in the landmark Stampede trial from the Cleveland Clinic. This is a randomized control trial of intensive medical therapy or sleeve gastrectomy or gastric bypass for those with type 2 diabetes. These included a range of BMIs, so including non-overweight patients. And you can see on the left, in both surgical groups, the hemoglobin A1C dropped dramatically. And even more importantly, on the right, this shows the number of medicines you're on to control your blood sugars. Those drop as well. And so the bariatric surgery can actually make your diabetes go away temporarily or even for good. So how does this work? So as I sort of alluded to, this was sort of designed with the idea that weight is simply calories in, calories out. You should just diet and exercise, and that's how you lose weight and maintain it. And so your gut really is just the cylinder. Food goes through it, it absorbs nutrients, and what doesn't get absorbed goes out, right? So if you make the surface of the area of the gut smaller, you can eat less, so maybe you're restricted because of just the physical size. And you eat less, you have less calories, and because there's less surface area going by, maybe you absorb less calories and more of it just gets dumped out in waste. So this concept of restriction and malabsorption. And these dietary changes in the weight loss are behind the effects of bariatric surgery. This is actually largely not true, and I'm going to try to show you some data that this idea of restriction and malabsorption is sort of outdated, and the surgery works through actually much more complicated mechanisms. Really one of the first indications that it isn't all about diet and weight loss was in this case study from Walter Pori's almost 30 years ago now. This is of a diabetic patient who came in and on the day of surgery had a blood sugar of almost 400, was on over 100 units of insulin. And then you can see sort of the trace of their insulin and glucose requirements over a couple days. And almost immediately after surgery, this patient went home with normal blood sugars and off of all diabetic medications. And so it showed that the bypass, before you even lose weight, within a day or two makes diabetes go away. And this is not just one patient. This is a study we did at the Brigham of all diabetics who underwent either gastric bypass or the sleeve, showing the number and types of medications they were on before surgery at baseline, and then highlighted in the box on the day of discharge, which is on average 2 days after surgery in this cohort. And you can see within 2 days of surgery, 26% of people no longer need medicines for their diabetes after a bypass, and a third after a sleeve. So there's something about the surgery that's happening right away, and this has got to be biology, right? So my lab studies the sleeve primarily. We use a mouse model. This is high-fat DIO mice, really common model, study insulin resistance. You can do a sleeve on the mice just like you can do in people. And in the sham surgery, we do a laparotomy and ligate the vessels, but don't divide the stomach. And these mice, just like humans, show improvements in glucose tolerance. And they also show elevation in a hormone called GLP-1 in their blood, glucagon-like peptide 1. Probably most people know it now as semaglutide or Ozempic, the medicines that are being used to treat diabetes and for weight loss, really a hallmark of all the bariatric operations. And relevant to diet, in the mice, you can control what people are eating, right? Some people, after they have the surgery, they eat less, eat healthier for a variety of reasons. In the mice, we actually feed them a high-fat diet before and after surgery, and the diet is exactly the same, yet they still have these glucose changes. This was work published a while ago by Dave Harris when he was a fellow in my lab, and when he developed a model where we did the surgery in lean, normal mice, and when you do on those mice, they actually don't lose weight. They have no changes in body composition by MRI spectroscopy, yet despite that, they show these same really impressive changes in glucose tolerance, sort of reiterating the effect that the surgery is doing something separate from weight loss and separate from diet changes. And when you put these in metabolic cages, other folks have shown this earlier, you can show a change in respiratory exchange ratio in these mice with an increase in the RER, so a tendency to burn carbohydrates right away after surgery. And there's an increase in glucose uptake, particularly in the fat depots in these mice. And again, this is independent of weight. So there's biology going on, which is taking out that piece of stomach that leads changes in metabolism globally in the mice. Dave is now on his own, has his own lab in the University of Wisconsin. He was kind enough to share some of this data, and he's been doing some more work, more analogous to what we've been talking about in this workshop, and I thought it might be interesting to share it. So he's doing the same sleeve model, this is in Western diet-fed mice, and then he randomized them to continue the Western diet or diets with different amounts of protein in them, and then did metabolic testing on them. And interestingly, in the protein-restricted group, they actually lost more weight. This is counter to what we do clinically. Clinically, we tell people to eat only protein. So he did the opposite and actually worked better. And they actually have improvements in fasting glucose as well. And so this sort of comes to the idea, and everyone knows this now, that the gut is really much more than just sort of a pipe that transmits food and absorbs nutrients, right? It's got an immune system, it's got nerves, it's got endocrine system that secrete hormones like GLP-1. And actually what's in the lumen is not just food and waste, it's actually nutrients, there's bile acids or microbes, and it's very complex physiology and complex organs going on here. And I'm going to show you some data that the interactions happening in the gut can affect what happens, particularly in the liver. So switching gears a little bit to the microbiome. Changes in the microbiome have been long associated with obesity. And bariatric surgery also has particular changes linked to various operations with overlapping changes that are conserved among the different operations. But whether these changes in the microbiome, like in a lot of things, are really association or causation in terms of obesity or surgery is really not very clear. So I'm going to tell you a bit of a story about surgery, the microbiome, and bile acids in particular. So just to remind everyone, bile acids are molecules made by the host and by the liver. They're not actually just bile acids. There's actually a big community of molecules with all these different alphabetical soup names listed here. And importantly, the bile acids are conjugated and chemically modified by bacteria within your gut to form secondary bile acids in particular. So there's already crosstalk between the gut microbes and your bile acids. And we'll get back to that. So this is collaborative work with Sloan Devlin, who's a professor at the Harvard Medical School, who's an organic chemist. And together we looked at bile acids in the gut after a sleeve gastrectomy, either in mice and also in fecal samples from patients who had undergone sleeve gastrectomy. At the time we did this, no one had really characterized individual bile acids in the gut. They had really focused on the blood. And she did this through detailed chemical methods. We surprisingly found one peak that was elevated post-sleeve, and she was able to identify that as a molecule called cholic acid 7-sulfate. And this is really the only bile acid increased in the sleeve, both in mice and humans. And worked together, we were able to demonstrate that CA7S, this bile acid, is an agonist for TGR5, which is a bile acid receptor in the gut expressed on L cells that leads to secretion of the GLP-1 molecule and improves glucose tolerance. And so you can treat mice with this molecule orally, and it will induce GLP-1 and it'll drop your serum glucose. So one way that sleeve can drop your blood glucose. What does this relate to the microbiome? So we treated these animals with antibiotics, and if you treat these animals with shotgun antibiotics and deplete their microbiome, you also deplete CA7S levels. So CA7S requires the microbiome. And then work I won't touch on here, but sort of a summary figure here, we went on to show the pathway by which the body makes CA7S. It's generated in the liver by a sulfation enzyme called SOL2A, and it gets secreted into the gut. And importantly, this whole process gets triggered by another bile acid, a secondary bile acid that's made by the gut microbiome called lipocholic acid, or LCA, that I'm going to talk about. And so if you do a microbiome transfer, you can reinduce this whole pathway and generate CA7S in a germ-free mouse. So I will touch quickly on some unpublished data around LCA. So LCA is this other molecule that's produced by bacteria in the gut. We had observed in our studies that said that CA7S was the only molecule increased, while LCA is the only bile acid molecule decreased after sleeve in the colon and in humans as well. And so we were curious, LCA is known to be a carcinogen, and we wondered, is it possible that the lowered LCA levels are also improving glucose control, meaning that LCA is bad for you, and sleeve helps you by dropping those LCA levels. So Yunjia, who's a postdoc in my lab, fed normal mice LCA, and you can see that this worsens glucose tolerance and increases their adiposity and worsens their respiratory exchange ratio. As I mentioned before, LCA is known to be produced by specific bacteria in the gut that express a specific enzyme complex called the bioperon. And in mice, the expression of the bioperon is dramatically dropped after surgery. In humans, the expressions also drop, as are the bacteria that make LCA. And so then we were interested in trying to figure out what is it about the surgery that leads to this reduction in LCA. So Snehal, who's a postdoc in Sloan Devlin, my collaborator's lab, took small intestinal contents, with the thinking being that small intestine's upstream of the colon, which is where the bacteria that make LCA is made, and put these on cultures from the cecum. And we found that if you take small intestinal contents from a sleeve and put it on bacteria, you can inhibit LCA production and the BICD expression. And sleeve itself increases TDCA, highlighted here. Sorry, going back, so we profiled bile acids in the small intestine. These are all the different bile acids in the small intestine. We found one molecule has increased, TDCA. So we then hypothesized that TDCA was the molecule that was driving down the production of LCA. And we showed that in vitro. And then in vivo, if you give TDCA, you can drop LCA production, reduce BICD expression, and improve glucose tolerance. And this is dependent on the microbiome because TDCA loses its effects if you then treat the mice with antibiotics. So TDCA is a good bile acid that's increased by the sleeve to drop bad bile acid production by the microbes. And going back to patients, TDCA is only in the small intestine primarily. This is hard stuff to get, so we can't get pre and post surgery small intestine. But what we could get was small intestinal samples from patients undergoing gastric bypass that were either diabetic or non-diabetic. And then we measured TDCA levels in those small intestinal samples. And we found that TDCA levels inversely correlated with diabetes control. So the higher the TDCA, higher non-diabetic patients had higher TDCA levels, and diabetic patients had lower TDCA levels, sort of confirming our hypothesis that TDCA is this good molecule that's increased by the sleeve. So I know we're short of time. I'll sort of breeze through this effect these last couple of slides. So I've shown some data around microbiome changes in bariatric surgery, and it's been known for some time, this is work from Lee Kaplan's group in Boston as well, that if you transfer the microbiome from a gastric bypass mouse, you can transfer some weight loss. And we've replicated that in my lab as well with the sleeve, but it's not really clear if you really need the microbiome. Is it necessary for the effects of bariatric surgery? So the way we tried to address this was by performing sleeve gastrectomy in completely sterile conditions in germ-free mice. So doing a sleeve in an animal that doesn't have a microbiome. So if you think the microbiome changes are necessary for the improvements in bariatric surgery, you should lose it in the absence of a microbiome. And I'll zip through this. This is technically quite challenging. You have to put everything in these isolators that remain sterile. This took this fellow here, Andre Moscalou, who's a surgical resident, about 3 years to do after multiple tries. But he was able to do this. You take germ-free mice, raise them completely sterilely, put everything you need to do the surgery and survive them and test them for 2 months afterwards in the isolator. We test them to confirm sterility. We took stool samples before and after and throughout the surgery to confirm that the stool is completely sterile. This took a few tries, but we were able to do this eventually. And we also confirmed that by 16S sequencing. And bottom line is this is the weight data, so the weight changes in the conventional mice on the right and the germ-free mice on the left under different diet conditions. The bottom line is the microbiome maybe contributes a tiny amount to the weight loss of a sleeve, but not much. Even in germ-free conditions without a microbiome, you still lose weight. The microbiome's a little bit more necessary for the improvement in type 2 diabetes. So under normal diet conditions, it's completely dispensable. The glucose improvement's completely the same under normal diets. But in the presence of a high-fat diet, you do need a microbiome to get the complete effects of the surgery. So this goes back to my summary slide again. Hopefully, had to breeze that pretty quickly, but hopefully convinced you that surgery isn't just about restriction malabsorption. It's actually changing molecules within the gut, particularly bile acids and microbes. And these communicate to the host itself to modulate secretion of hormones like GLP-1 that then affect diabetes and the weight. Thank you again for having me, and these are all the folks who did all the hard work here.
Thank you, Dr. Xu. In the interest of time, I think we should approach Dr. Xu at the break for any questions. It's my distinct pleasure now to introduce Dr. Chris Hahn. He's an associate staff and associate professor in the Department of Cardiovascular and Metabolic Sciences at the Cleveland Clinic. After earning his PhD in Rochester, he actually was Jay Mitchell's postdoctoral fellow, and that's where I first met Chris, who was a superstar and helped out training some of the postdoctoral fellows that worked in my lab. I liked him so much, our department put together a multi-million-dollar package to keep Chris in Boston at the Brigham. But he went back towards his hometown area in the Midwest, and the Cleveland Clinic's been blessed to have him since. Chris is going to talk to us about inhibition of the mammalian transsulfication enzyme and microbial metabolites. Chris.
Thanks a lot, Keith. That was very, very kind of you. So I'll start this talk with a question, and I'd like you to raise your hand. How many in this room would like to live a long life? I raised. All right, awesome. So largely due to improvements in public health initiatives, such as vaccines, antibiotics, and sanitary engineering, your chance of living a longer life is improving. So in the year 1990, only 6% of the world's population was over the age of 65. If we can go by decade, by 2020, 10% of the world's population was over the age of 65. And predicted in the future, by the year 2050, over 16% of the world's population is over the age of 65. So we're on the right track. But living longer doesn't necessarily mean living healthier, because with old age comes aging-related morbidities, such as brain and cognitive decline, cardiovascular disease, problems with immunity and increases in inflammation, and last but definitely not least, increases in cancer, oncological diseases. So how do we deal with these aging-related morbidities? So current medical strategies are, we have a drug for cancer, we have a drug for heart disease, we have a drug for diabetes. And if we keep following this pattern and we improve those individual drugs, we'll have this scenario right here, scenario number 2, in the sense that we might actually extend lifespan, but not necessarily extend health span. So this is actually not a great scenario. We're living longer, but not necessarily healthier. Another approach is an approach called geroscience, where these innovative geroscience approaches can be applied to when humans start aging, around the age of mid-30s, we start to age. And in doing so, we don't necessarily improve lifespan all that much, although you do a little bit, but the main goal is to improve health span, in the sense that the amount of time we live without disease or without these aging-related disorders. So the questions we ask in the geroscience field are, what are these experimental geroscience approaches? What are their mechanisms of action? And likewise, can they be leveraged for the prevention and treatment of these aging-related morbidities? So it's been talked about quite a bit today. So one of the most well-studied geroscience intervention is dietary restriction. Dietary restriction is an umbrella term encompassing caloric restriction, so daily reduction in overall calories, not really playing with the ratios of protein, carbohydrate, or fat, intermittent fasting or time-restricted feeding, and then likewise getting more mechanistic, such as protein or essential amino acid restriction, and has been also brought up today, sulfur amino acid restriction, such as methionine restriction, has been shown to be sufficient in itself to extend health span and also lifespan. Another geroscience approach, this is mainly more experimental, especially in mouse models and worm and fly models, are looking at endocrine manipulation. And in mouse models, we can think about hypopituitary dwarfism. This is where you have a reduction in growth hormone and thyroid hormone signaling, also growth hormone receptor knockout mice, the world record holder for mice over 5 years of age, created by John Kopchik. And then in the end, most of these models have a decrease in IGF-1 signaling. So it's kind of an anti-cancer environment. And definitely last but not least, but probably the easiest to utilize for humans are pharmacological interventions for extension of lifespan. And so some of the drugs here, rapamycin, metformin, and acaribose, with rapamycin being, hitting the target of rapamycin, mTOR, and then metformin, acaribose, usually being used for anti-diabetic treatments. It should be noted here, we'll get to this later in the talk, that a lot of these drugs are actually bacterial metabolites. And so the interaction between, bacteria and what they produce in our own health is inherent for both our lifespan and our health span. And so again, we ask the questions, Are there common molecular pathways impacted by all three of these types of geroscience approaches? And can we focus on that pathway and leverage it for aging-associated morbidities? And so from the work of Jay Mitchell's laboratory, Vadim Gladishev's lab has also studied this as well. The central pathway for a lot of these geroscience approaches is on the transsulfuration pathway. And a particular enzyme in the transsulfuration pathway, cystathionine gamma lyase, abbreviated here as CGL. So the transsulfuration pathway, you take the essential amino acid methionine, so there's that sulfur on the methionine. Through multi-step reaction, it gets converted to homocysteine. Homocysteine is acted upon by an enzyme called cystathionine synthase, which is the PLP-dependent enzyme. PLP is the bioactive form of vitamin B6 to form cystathionine. Cystathionine then gets acted upon by CGL, again a PLP-dependent enzyme, to form non-essential amino acid cysteine, and you have there the sulfur on there. However, it should be noted that CGL is actually quite promiscuous in terms of the reactions it brings in and the products it makes. In the sense, you can see here that cystathionine, or CGL, can actually catabolize cysteine and homocysteine to produce a number of products. And one of the byproducts of these reactions is hydrogen sulfide gas. Hydrogen sulfide gas, at least in biology, acts in three main ways. One, it acts as an antioxidant. So protecting against oxidative stress, of course, would be a direct way to kind of slow the aging process or slow the damages of aging. Next, it can modify proteins through a process called persulfidation. And persulfidation, or also called sulfhydration, is a post-transition modification that occurs on cysteine residues where you can form these long polysulfide tails. And it's thought that this long polysulfide tails can protect a protein from irreversible oxidative stress in that cysteine, or like other post-translational modifications, it can modify the protein structure and ultimately its function. And last but not least, hydrogen sulfide can modify mitochondrial bioenergetics. So if levels are low enough and not toxic, it feeds electrons into electron transport chain to make ATP through a complex called sulfite quinone oxyreductase, abbreviated here as SQRLDL. However, if levels are too high, it can actually inhibit mitochondrial respiration by inhibiting complex 4, much like cyanide. So I don't want anyone going home and sniffing hydrogen sulfide, okay? It's better if your own body produces it. It could be deadly if you get too much. So to be safe, just don't take it from the external sources. So unfortunately, conversely, in terms of these geroscience approaches enhancing CGL and H2S production capacity, as we age, we published this here about a year and a half ago at the chair of my department, Stan Hazen, measuring sulfide levels in human patients. We measured 3,102 human patients. There's a significant decline in both men and women in the circulating sulfide levels. So it might be important for us, in terms of living longer and healthier, to counter this age-related decline and boost our own production of this gas. And so historically, individuals would seek out these sulfide pools and swim in these sulfide pools. It really wasn't effective. It wasn't a fountain of youth. So in my own laboratory, especially with Jay's laboratory, we found, what are some effective interventions? So dietary restriction through activating GCN2 and ATF4 can stimulate the transcription of CGL. Likewise, thyroid hormone, when bound to the thyroid hormone receptor, are repressors of CGL. So undergoing a hypothyroid or subclinical hypothyroid state can boost CGL expression. This increase in CGL leads to an increase in H2S production. And what we found and published and also in preliminary data, I'm not really going to go into it too much, is that this is necessary and sufficient to promote extended and low tumor burden longevity in mice. And likewise, we've also published 2 papers in the last couple of years that this endogenous production of H2S, by stimulating it, actually acts as a suppressor against really devastating brain cancer, glioblastoma, in mice and in human cell culture models. However, I'm going to go on a new angle here that I really haven't talked about, and really only touched about it briefly in my laboratory, but I think it's quite interesting. Are there diet and microbiome-related enzyme level regulation of CGL that can be leveraged? Such as there diet-related enzyme level regulation of hydrogen sulfide production, or is there diet enzyme level regulation of H2S oxidation and removal? And so first we'll start on the first one in terms of, can CGL be regulated on an enzymatic level through gut-related metabolites? And so here we have the crystal structure of CGL. Here's a tetrameric form that's not bound to vitamin B6. You can see it's kind of an open conformation. And when it's bound to vitamin B6, it has a conformational change. So there is flexibility to this enzyme that we think that could be leveraged for its regulation. Known inhibitors of CGL, So one of them is a chemical called propargoglycine, abbreviated here as PAG. This is a specific suicide inhibitor CGL via covalent binding to the vitamin B6 site in the enzyme. Likewise, there's a chemical called AOAA, which is a universal inhibitor of all B6 utilizing enzymes. So there's probably at least thousands of them. So that'll also include CGL. We'll focus a little bit more on the PAG because it is specific to CGL. And so when we were studying the glioblastoma, we were looking at a variety of glioblastoma cells in culture. So this KR158, GL261, and CT2A, these are glioma cell lines. We grew the cells either without PAG or with PAG. And we then overlaid the plates with a piece of paper that has lead acetate baked into it. And so if hydrogen sulfide interacts with lead acetate, it forms lead sulfide, which are these nice kind of brown or black precipitates. So the darker the circles means more hydrogen sulfide is produced. So you can see that PAG was quite effective at diminishing the production of that gas. So how did it affect the cell growth? And what we saw was by inhibiting endogenous production of that gas, it made those glioma cells grow much more quickly in culture. In all three of those cell lines, they grew quicker. So we published this a couple of years ago. However, the question is, where does PAG come from? I think it's often neglected in the field. And it was actually first identified in the 1940s as a metabolite coming from a Streptomyces soil bacterium. In the sense, these Streptomyces produces propargoglycine, and they will inhibit the growth of other bacteria and fungi in the surrounding area by inhibiting their sulfur-containing amino acid metabolism. So it's almost like an antibiotic these Streptomyces were producing. So we asked, are there novel regulation of CGL via the diet and microbiome-related metabolites similar in size and structure as PAG? And could the impact of these gut-related metabolites on CGL provide their causal impact on health and disease and cardiovascular disease and cancer? And so to start, we took 2-month-old female C57 black 6 mice. We put them under 2 weeks of a very strong antibiotic cocktail, neomycin, ampicillin, metronidazole, and vancomycin, to kind of really wipe out their microbiome in their entire body. After that 2 weeks, we then harvested the tissues and performed hydrogen sulfide production assays and protein persulfidation assays. And you can see here in the kidney and the liver, the animals under the antibiotic treatment had increased production of that hydrogen sulfide gas. So kind of wiping out that microbiome boosts the endogenous production capacity of that gas in those tissues. Likewise, looking at the signaling, the persulfidation signaling, we did through a biotin-thiol assay coupled with mass spectrometry, we see a really nice enrichment for protein persulfidation in the kidney if the mice were on antibiotics. Likewise, also enrichment for persulfidation in proteins in the liver if the mice were under antibiotics. So what could potentially be the microbial metabolites produced by the gut that could be inhibiting H2S production? And so again, I come from the department of Stan Hazen, who's previously published before that diets high in protein, especially diets high in red meat and dairy, cheese, and eggs. There's a lot of choline and carnitine in these diets. That when we eat these, they travel into our system in terms of the choline. The choline then, or the carnitine, gets broken down by the gut flora, so the microbiota in our guts, to form trimethylamine, abbreviated here as TMA. Trimethylamine is quite volatile, circulates throughout the body, and then the liver gets converted by hepatic FMOs to trimethylamine and oxide, FMO, or TMAO. And then TMAO has been shown to be at least correlative in humans and causes in mice to cause for atherosclerosis and hypertension, while on the opposite, hydrogen sulfide is a vasodilatory mechanism. So we really looked at, can these metabolites based off of this pathway be impacting CGL activity? And so we took liver homogenates from wild-type mice, mixed it with PBS, 1 millimolar PLP, so the vitamin B6, and plus or minus 1 millimolar of the metabolite, either the TMA, TMAO, and betaine. And what we saw was a moderate but significant reduction in liver H2S production capacity if they were incubated with these gut-related metabolites. Importantly, if we incubated 1 millimolar of the precursors, the choline and carnitine, there was no repression, indicating it's much more specific to the actual microbial metabolites as opposed to the precursors that we bring in the diet. And just to see if, of course, 1 millimolar is quite high. Is it just kind of gunking up the system? We took livers from CGL knockout mice, again, incubated them with the metabolites, and we did not see repression, indicating that these metabolites at 1 millimolar aren't impacting CBS or 3-MST, other H2S-producing enzymes, or interacting with vitamin B6. And so then we looked at purified human CGL in vitro systems, and again, incubated with much lower concentrations of TMA, TMO, and betaine. And again, what we see in some of these cases, even at 5 micromolar, we can get a significant reduction in enzymatic production capacity from TMA and TMA and betaine. And again, using more biophysical techniques to look at, are they actually interacting with CGL itself or vitamin B6? So in terms of looking to UV vis spec with PLP, you get a peak here around 390. If you incubate PLP with AOAA, the universal inhibitor of anything that uses B6, we get a shift in the spectrum to around 330. And if a PAG, which interacts with the enzyme itself and not the B6, we don't have a shift. And so we saw when we incubated TMA, TMO, and betaine with vitamin B6, we do not get a shift. Again, suggesting that these molecules are interacting with the protein itself, not the cofactor. And so then we followed it up with more biophysical techniques such as thermophoresis, where we incubated CGL with known suicide inhibitor PAG, and we get a shift of 15 units. Again, if we incubate CGL with TMA, we get a shift of 9.2, and we incubate CGL with TMAO, we get a shift of 4.2. So they're on the very similar range as we get with that known suicide inhibitor, indicating a direct kind of chemical protein interaction. We then also used plasma resonance tests to get very similar results, showing that these are interacting with CGL. And importantly, if we pre-incubated CGL with PAG, this dampened the interaction with the TMA and TMAO with the CGL, indicating they're potentially binding in that same pocket as the PAG, which is a known pocket. So in summary, we were talking about the production angle. What about the oxidation and removal? So how mitochondria interact with bacteria? Mitochondria were once bacteria, so they're cousins basically. So they will signal to each other. And so in terms of the oxidation and removal of H2S that occurs in the mitochondria, mainly through a complex called sulfide quinone oxyreductase SQRDL, which produces this glutathione persulfide, is then acted upon by ETHE1 to form sulfates, sulfites, and thiosulfates. And what we can see in the realm of glioblastoma is that there's an increase in SQRDL expression in the glioma compared to the non-cancerous tissue. Same for ETHE1 and the other two enzymes that are involved in the detoxification and removal of H2S from the cell. So under glioblastoma, we see there's usually a decreased production capacity of H2S gas, but now we're also seeing an increased capacity to remove it. So glioma do not want H2S around. Also looking at the graded glioma, looking at SQRDL expression, ETHE1 expression, we see as we get much more to the lethal forms of glioblastoma, expression of these H2S removing enzymes is increased. Survival, those with higher levels of SQR and higher levels of ETHE1 are shown here in the red. These are glioblastoma patients, have a lower chance of survival than patients with lesser expression of those sulfide removing enzymes in the tumor. So are there inhibitors of SQRDL, and can they be used for GBM therapy? And is there a link to bacterial metabolites that regulate mammalian SQRDL? And so luckily, there is. There's only been one paper published on them. They are synthetic, but they are proposed to inhibit SQRDL. And so here we have a variety of cell lines with over, these ones here being glioma cells. We did Western blots of SQRDL, ETHE1, of course, the loading control. And what we went for is the glioma line that had the highest expression of this SQR and ETHE1. That's the KR158. And so if we incubated the KR158 with a vehicle control, PAG, so we have a reduction in H2S production. And if we put in those supposed SQR inhibitors, we don't really get a response all that well in these mouse glioma cell lines from the SC904, but the other one, this HTSO7545, you get a really nice boost in H2S production, indicating you're getting an inhibition of SQR. And so looking at the cell growth, it mimics this. In the sense that the inhibitor that did nothing, those cells grow just fine. But if we look at the growth at various doses of the HTSO745, we see we can reduce glioma growth in culture. Now, what about human glioma cells? What's really interesting is you remember that this FC94024 did nothing in the mouse? It's extremely lethal in the human glioma lines. You see here that any dose to be used wipes them out. While the drug that was effective in the mouse cells is still effective in the human ones, it's HT705. You can see the growth patterns here, especially for this FC9204, extremely lethal in the human glioma cell line. So again, moving forward, we're going to need some help from the microbiome community, really identifying bacterial metabolites that inhibit SQRDL similar to our synthetic models right here. And so in conclusion, so this again, it's a work in progress. We're proposing that dietary restriction in of itself can elevate CGL expression on a transcriptional level, but also dietary restriction will lower our circulating levels of TMA, betaine, and TMAO, which then serve, as inhibitors. So if we can remove these, we remove that suppression. And by boosting up H2S and boosting up persulfidation, we're seeing maximized lifespan in mouse models, maximized health span in mouse models, and also tumor suppression in glioblastoma. And so, again, if we can do some microbiome transplants of H2S, producing bacteria, that might be beneficial. Likewise, if you can identify bacteria that will produce these metabolites that can inhibit SQR, then that will relieve that suppression and increase H2S. So again, and also in honor of Jay and also in honor of Keith, my interaction with them, how do you bring this to the clinic? And so in terms of treating people with glioblastoma, it's extremely devastating. I have friends, I've had family. You get diagnosed, and within about 12 to 16 months, it's fatal. Usually people, you don't survive glioblastoma. So you have the option of dietary restriction. But usually in the clinic, especially patients with glioblastoma, physicians aren't going to prescribe a dietary restriction. So if we move to another model of modulating endocrine activity, especially looking at hypopituitarism. And so right now we have a clinical trial that started last April. It's in collaboration with David Pierboom, who is a glioblastoma expert, MD, at the Cleveland Clinic. And so for recurrent glioblastoma, we have patients undergoing methimazole treatment to lower thyroid hormone levels. So then that will remove that suppression of H2S. The H2S acts as the tumor suppressor against the glioblastoma in combination with standard of care temozolomide. And so again, very briefly, this is our update we have right here. So it's working. In the sense that the glioblastoma patients undergoing the methimazole, we see here, so this basically, here's your sex match, age match controls. And as they progress in the treatment, we see between a 1.5 and a 3-fold increase in the circulating salt H2S production capacity. And this is all reaching statistical significance. So hopefully this also, improves the survival of the patients and also their quality of life. But at the very least, from these very preliminary data, by taking the methimazole, it is enhancing the production of the gas. And so with that, I'd like to acknowledge current members of my laboratory that worked on this project, past members that worked on the project, collaborators on the brain tumor side of things, all the funding, and last but not least, acknowledgments to James Mitchell, Mitchell and Ozaki labs again for, really instilling in me, a passion for science and really looking how do you make the connection between bench to bedside and how do you go from bedside back to bench. Thank you.
Thank you, Chris. And I'd like to sort of take the honor and privilege of the floor for a few minutes and get our organizing committee and the local organizing committee to come to the stage. Zafer, Furkan, and Turgay, and Atta. Can you just come up? So we hosted this meeting in Boston a couple of years ago, and Lorraine is here with me, and they can attest to the amount of work it takes to do an event like this, and it's been fantastic. Atta, I don't see right now. He may be in the operating room taking some pictures. Yeah. But anyway, I just have some gifts from the group in Boston. We passed on the torch to you. You guys handled it very capably. For you, and then we'll give this to Atta. Thank you. And then this is for you, Professor, and of course your lovely wife. Thank you. I know it was a team effort, but again, much gratitude from the whole team in Boston and beyond. So thank you.
We appreciate for coming here. Really enjoyed with being with you. Thank you very much.
Yeah, so we'll have a 10-minute break. So we're getting into the next session in 10 minutes. All right. Sorry to take that.